Thank you everyone for joining us today. Our webinar today is about the sperm selection with Dr. Dave. We'll hand over to you, Dr. Dave. Thank you.
Thank you, Emma. I'll just share my screen. Just bear with me. Hopefully you can see that. Okay, so today we're going to talk about sperm selection. Thank you to Amir and Hadil for asking me to give this talk. And what I want to talk about is how we select populations of sperm for use in treatment and how we can select individual sperm when we're looking to inject them. So we'll take a quick look at sperm preparation techniques and now how that impacts the sample. We'll also briefly touch on how the preparation technique impacts treatment outcomes. Briefly talk about the conditions for sperm, and then look at the selection of individual sperm for use in treatment. The first thing to say, of course, is that the sperm structure mirrors its function. So that the purpose of sperm is to deliver the male genetic material to the oocyte and drive embryo development. And it does that by making this unique structure during spermiogenesis from round spermatids to the mature spermatozoan that we are used to dealing with the lac of cytoplasm and the active flagellum. Now, the delivery of these spermatozoa depends on the ejaculate. And the ejaculate is comprised of a range of important elements, including seminal plasma, non-sperm cells, debris, and it can also carry bacteria and viruses and so on. And of course, it's primarily designed as a sperm transport medium, getting the sperm from the man to the female genital tract. And what we want to do during treatment is actually get rid of all of the other bits and isolate these mature, motile, functional spermatozoa. And we do that through a range of sperm preparation techniques. So I'm going to take you through a couple of those and talk about the principles, but also hopefully highlight some of the key factors. Now, as I've said, we want to collect the mature motile sperm. So we're looking to isolate a selected population of sperm with normal morphology, however we define that, minimal DNA damage within the nucleus of the sperm. And of course, because the sperm's function is dependent on interaction between the sperm membrane and that of the oocyte, we need to make sure that those cell membranes are intact and have functional binding proteins. And what we want to do during the course of sperm preparation is get rid of the things that will inhibit or detrimentally affect the fertilizing ability of sperm. And that includes seminal fluid, which plays an important role in sperm transport, but actually prevents normal fertilization. We want to get rid of the non-sperm cells because we know that they are a source of reactive oxygen species, and hopefully you're aware that reactive oxygen species and oxidative stress is particularly damaging to sperm. And we also want to remove the immotile sperm, the ones that have no functional properties that are useful for use in treatment. And so what we're aiming to do is get this highly selected population. You can see here a picture of a failed to fertilize human oocyte where the zona pellucida has been expanded, and we've stained up the cells, and you can see the sperm adherent to the outside of the zona, and they show a much, much better morphology than you see typically in a prepared sample and certainly in the raw semen. So it's clearly morphology based. And what we also want to do during our sperm preparation techniques is actually prepare the sperm for capacitation so they they acquire that ability to fertilize the oocyte, whether that's in vitro during IVF or in vivo if we're performing intrauterine insemination. And what we want to do is avoid side effects such as a potential to introduce infection, and also because of prostaglandins in the seminal fluid, we want to avoid uterine spasms caused by the introduction of that. So we want that motile, normal subpopulation of sperm. And there's a few techniques. I'm going to concentrate on a couple of them, the more common ones. And the first one is, of course, the swim up. So no surprise here that selection during a swim up is based on the ability of sperm to swim out of seminal plasma into an overlying medium. And therefore, because it's motility based, it generally is reserved for good quality samples where the motility is good. It's an easy technique, with the caveat that it demands a certain amount of technical finesse, because poor technique allows some seminal plasma to be dragged up, and that can impact the quality of the sample that we get at the end. And a really nice paper from Lars Bjorndal and colleagues showed that if you use zinc as a proxy marker of contamination by seminal plasma, swim-up samples often have elevated levels of zinc, showing that seminal plasma is dragged up or taken up during the harvesting process. So we have to be mindful of that. What we know is swim up is effective at isolating motile, morphologically normal sperm, and it reduces the proportion of those sperm carrying DNA damage, and it reduces the risk of reactive oxygen species being carried over. The general guide here is that you should always do a direct swim up from whole semen, never wash, then swim up, because if you wash first, you're concentrating the good sperm down with the non-sperm cells and dead sperm that can cause damage. So this is a very simple, easy technique, generally low yield, but highly motile sample at the end of it. Now, the other main approach is to use a density gradient. And I think it's important to start off with some of the basics here because it's often overlooked. Here with a density gradient, we're using media, usually a colloidal suspension of silane coated silicon particles. And the principle here is that we select based on the density of the sperm. And it's this principle that the cells will separate based on reaching a point of equal density in the column. And that's called the isopycnic or buoyant separation approach. Now, because it's a colloidal suspension of silane coated silicon, the density gradient media exerts no osmotic effect above and beyond the base medium. That's different in some density gradients that have been used, such as sucrose gradients and Ficol. But what it does is it allows us to pick out the most dense sperm, and because of the loss of cytoplasm, the most mature and most motile sperm tend to be the most dense. So we collect them at the bottom of the tube. So that's the guiding principle of all of the density gradients. It's worth noting as well that again, because they're a colloid, these are not viscous samples. They're dense, but they're not viscous because it's a colloidal suspension, not a solution. So here you see the sort of standard density gradient where you have a couple of layers with the semen on top and you centrifuge and then harvest the pellet from the bottom of the tube. So we're selecting a high yield, highly motile population of sperm, and it's motile even though we're selecting based on density. Now, we know we can play around with the numbers of layers of gradients, the speed of centrifugation and the time of centrifugation, but it's worth noting that sperm generally reach their isopycnic points, that's the point of neutral buoyant density, after centrifugation at 300 G and 20 minutes. So really, spinning them harder and faster is not likely to do them any good. And our recommendation generally is to set up more tubes to increase yield rather than spinning harder and longer. It's a bit more labor intensive than the swim up, but it gets good results, and it's generally the go-to technique if you're dealing with virally positive samples. Now, the other thing to say about density gradients is typically they have all been at the same osmolality. So if you spin down, semen is typically somewhere between 320 and 340 milliosmoles per kilo. And therefore, if your gradients are isotonic, typically at 270, 280, there's a degree of osmotic stress as they go through the medium. And you can overcome that by actually producing an osmotic gradient in your media, as well as a density gradient. So for example, in the Origio gradients, we go from around 320 in the semen to 317 to 333 in the 40% layer, and then down to 297 to 313 in the 80% layer. So it's more of a stepwise approach in osmolality before you put them into the wash medium. Now, the thing often leveled at density gradients is that centrifugation is a problem. And I would suggest you trawl the literature, and as I have done. And what it will tell you is that actually the data on centrifugation shows that you can see a decline in sperm motility when you spin at high speed. And at high speed, we're looking at well above 500 G. So this paper by Mackler and Jacobi from way back in 1980 showed that the decline in motility was seen if it was over 580 G, but at typical spin speeds that we use in the lab, it wasn't seen. There's also some evidence that if you go up to 600 G, you see a drop-off in DNA integrity that you don't see in a swim up without centrifugation. And there have also been a slightly more recent paper suggesting that centrifugation steps at 400 G can impact acrosomal loss, but it doesn't alter fertilization and cleavage. So the principle here being that if you are conservative with your spin speeds, there should be no particular harmful effect. And that's borne out by this very recent paper by Quinn and colleagues, where they did a randomized controlled trial comparing centrifugation and density gradient with a microfluidic chip, and showed that the outcomes in terms of embryo quality and clinical pregnancy and so on were unaffected. Note that they did use a low spin speed, and that's the take-home message. Now, if we think about microfluidics, because these are very interesting ways of dealing with sperm, and they do avoid or can avoid centrifugation, there's a number of different approaches to microfluidics: passive, active, and so on. And if we look at the standard sort of microfluidic chips, these rely on a sort of passive approach to microfluidics. And this really relies on the fact that sperm accumulate at boundaries, and there's a change of swimming pattern within a micron, where sperm change to this sort of slithering mode, which is more flattened type of motility, and they generally move considerably faster along boundaries. So that's really how these chips work. Now there are other approaches. So the more active approaches, you can have microfluidics where they use chemical attractants or a thermal gradient, a temperature gradient. More commonly, it's rheotaxis, so it relies on motility flow. And again, really interesting approaches to separating the best sperm. But it's worth noting this most recent paper, Zhang from 2016, also suggested that rheotaxis it appears to be quite a passive process. There's no direct evidence of different flagella patterns in the selected sperm above those that are not selected. And there's no evidence of calcium influx or active signal transduction. Very effective methods of selecting sperm, but we're not quite sure exactly how much different the population is compared to standard methods. Now, there are a whole range of other techniques to separate the population of sperm for treatment. You can use migration sedimentation chambers. You can use the charge on sperm to select them by producing a positive charge on a surface, and the best sperm are negatively charged. You can use things like MAX and Nexin-5 binding. They all have their place. They are in relatively modest use if we look globally. So I'm happy to talk about them more in questions, but I'm going to move on for now. What do these sperm selection techniques, sperm preparation techniques, do to the sample itself? So the absolute guiding principle of all of these techniques is that you end up with a more motile, more morphologically normal, and more motile, more morphologically normal sample with better DNA integrity. And actually, if you look through the literature, that's largely borne out by a lot of the studies. So this is a one from 2021 from Rad and colleagues, just doing a comparison of swim up and density gradient. And what it shows really nicely is that if you look across the board, swim up and density gradients will reduce the level of DNA fragmentation. They have an impact on the proportion of sperm with unreacted acrosomes and so on. But the overall message is that you can see in different studies some slight differences between these techniques. Some will suggest a swim up is better than a density gradient. Other reports show the opposite. The overall picture seems to be that if you do a good quality sperm preparation by your chosen method, you will be improving the morphology, the motility, and the DNA fragmentation, and they can work pretty much equally well, I think. Now, there are some caveats to that. The first caveat, and this is a paper by the EVI group, Masegwe and colleagues, from quite a number of years ago here, where they were actually looking at the relationship between outcomes and the oocyte quality in egg donation cycles. But what they, interestingly, as a side effect, what they found was that when they looked at sperm DNA fragmentation after swim up, you can see the usual thing that the sperm DNA fragmentation in the raw sample is relatively high, and it's drastically reduced after the swim up. That's consistent with all of the other studies. What's really interesting is in this study, what they showed is that in the frozen thawed sperm, first of all, the DNA fragmentation was higher after freezing and thawing compared to the population in where they were using fresh. Not sure they were the same population, so just not sure that's a genuine finding. But what is really interesting is that when they prepared frozen thawed sperm by swim up, the reduction in DNA fragmentation is far, far lower than it is for fresh samples. So the impact of the swim up in reducing DNA damaged cells is far less effective when you're using frozen thawed sperm. So just keep that in mind. The other thing to say is that in doing all this good work to reduce DNA fragmentation by doing a really good quality preparation of our samples, we can partly undo that by handling the sample badly after and before preparation. So this is some data from two papers by Nabi in 2014 and Matsuura from 2010, just demonstrating that if you look at neat raw semen samples from the time of production to the time of preparation, if you leave them for any length of time, the DNA fragmentation is likely to increase, and it's likely to increase more if the sample is kept at 37 degrees. So the message there being, if you are preparing sperm samples, ideally prepare them as soon as possible after preparation, and ideally no longer than 30 minutes between production and preparation. And if you can't do them immediately, then make sure you're not leaving them at 37 degrees, because the rate of DNA damage will be higher. Similarly, if you look at post-preparation, you can see the same thing. So if you leave samples after preparation and before use, the DNA fragmentation will slowly increase at a spontaneous rate of DNA fragmentation, which is lower at room temperature than it is at 37 degrees. So again, after preparation, it's best to keep your samples at room temperature and just warm them up before use if you're doing an IVF, for example. This particular study from Rougier and colleagues from 2013 actually compared men with low levels of DNA damage in their neat sample and those with high levels of DNA damage in their neat sample, and then looked at the rate of DNA, spontaneous DNA damage caused when they are kept in either Hyaluronan or PVP, so in sperm slow or PVP. And it shows the same thing, that over time, up to 90 minutes, you get this significant increase in DNA damage. So again, just be careful with your samples before and after preparation. So what do those preparation techniques, what does it mean in terms of outcomes for treatment? Well, if you look at the WHO manual, it essentially suggests that you should use a direct swim up for normal samples with good quality motility, good numbers of sperm, and then the density gradient is more suited, but can be used for all samples, but it should be used for the suboptimal samples. But ultimately, the choice of method actually has no clear impact on the outcome. So this is a well-cited paper from Boomsma and colleagues from 2019, where they looked through a systematic review of outcomes for intrauterine insemination and could find no difference between a swim up and a gradient in terms of the pregnancy rate per couple, or indeed the miscarriage rate per couple. So it didn't seem to have a big impact. Again, with the warning that we don't have a lot of great data. There's a lack of large, high quality randomized controlled trials. So the conclusions are a little bit uncertain. Plus, of course, they didn't have access to live birth data. Now, perhaps more interestingly, if you look at the IVF and PIXI data, and this is a paper just from last year from Rao and colleagues, where they looked at cumulative live birth rates after IVF and PIXI, again with density gradient and swim up as the two treatment groups. So this was a retrospective study using a propensity score matching analysis. But the bottom line is, if you look at the p-values, there was no difference between the two techniques in terms of fertilization rate, the good quality embryo rate, blastocyst rate, cumulative live birth and live birth per transfer. So the bottom line is the treatment, the way you select your population of sperm appears to have little impact on the outcomes. Now, in thinking about how we select populations of sperm, it's worth just quickly mentioning how we control conditions that those sperm are handled in. I've already mentioned that temperature is important for spontaneous DNA fragmentation. We also need to think about the media that we use. And what we know from multiple sources is that sperm like different conditions from those we typically use for our oocytes. And that's perhaps not surprising. Sperm are doing different things. They need to capacitate. They need to undergo the acrosome reaction in order to facilitate fertilization. And that's all an amalgamation of processes in the sperm that are required to acquire that full fertilizing ability. And what sperm will need to do is the head of the sperm undergoes changes. It's a very fluid membrane with cholesterol patches that allows fenestration during the acrosome reaction. That allows the release of enzymes from the acrosomal cap. And similarly, the tail also undergoes changes. So it acquires hyperactivation, which, as you probably know, is really important for the way sperm interact with membranes and ultimately drives through things like the vestments of the oocyte, the cumulus mass and so on. And all of that is dependent on the environment around the sperm. And what we can summarize that as is that sperm need a reasonable amount of albumin. They thrive in the presence of higher levels of bicarbonate, and they also like higher levels of alkaline pH than we typically see for our oocytes. And you can see that this is a fantastic paper from Achikano and colleagues from 2018 that showed how if you increase external pH in around the sperm, so in your handling medium, that drives a higher intracellular pH, which also allows calcium influx, a higher level of intracellular calcium, and that's critical to this acquisition of hyperactivated motility, which is so important. It also showed that motility is dependent on higher levels of bicarbonate. And we've done similar work in combination with Matt Tomlinson at the University of Nottingham and his master's student, Natalie De Rosa, where we did work on a range of different media, of different compositions, different pH, different bicarbonate, and showed that high levels of bicarbonate, high pH, were much more supportive of sperm motility. And that's important because it drives what we do. And you can see here, again, just taking our range as an example, that we have some differences from some other media in as far as our media are at 8 to 8.5, so considerably higher pH than we would have for oocytes and embryos. I've already talked about the osmolality gradient for the preparation, but you'll also see that we've got generally higher protein in the wash medium compared to other elements. So Origio products tend to use 5 milligrams per mil of albumin. The sperm wash uses 10. And you can see that there's a high level of bicarbonate as well as the requirement for antioxidants. And we know antioxidants are particularly useful for sperm, where it's much more evident, for example, than it is for embryos. And taurine in particular is a well-established antioxidant for sperm media. Okay, let's talk about how we select individual sperm. So we've got our population, we've got better motility, we've got better morphology. and we've got lower DNA damage. But when we come to selecting individual sperm for ICSI, the guy at the microscope is basing it on motility and morphology. And we know that there are some problems with that, because we know, for example, in oligoasthenoteratozoospermic men, so the men with low counts, motility and morphology, their morphologically normal sperm had a much higher rate of aneuploidy and DNA damage than normal sperm of normozoospermic men. So even though we're picking what look like normal sperm, they still carry a higher rate of defects. So we've looked at different ways of selecting sperm. IMS is one. So this is important because it uses morphology, and you'll be aware that we use these high magnification imaging systems, and essentially looking at nicks in the sides of sperm and vacuoles and so on. And we can try and pick out the ones that are morphologically normal, the ones that show no vacuoles and no nicks. Now there's variable reports of the efficacy of IMSI. The same group produced two systematic reviews, with the later one suggesting that actually sperm selection in selected patients can help overcome late paternal effects, so blastocyst formation and implantation. So there's still a role for IMSI if you look for targeted groups. Now, a more interesting observation is this one from Berkowitz and colleagues from 2018, where they looked in some detail at the presence and location of vacuoles, and interestingly showed that it was less around the number and size of vacuoles as we've previously used for IMSI classifications. And what they suggested is that the genomic instability that you see in sperm is more related to the location and depth of vacuoles. And they related it to the copy number variation in the sperm, and suggested that if you get low copy number variation, they have no vacuoles, or they're not deep inside the sperm head, whereas the ones with high copy number variation have them in particular areas like the equatorial segment, or they're deeper into the sperm head. So we might need to rethink our classification for IMSI in light of evidence like that. Now, the other way of looking at sperm is to use hyaluronan binding, and you're probably aware that hyaluronan is the principal extracellular component of the cumulus mass. Sperm bind to it naturally during penetration of the cumulus, and a whole range of studies have demonstrated that the ability to bind hyaluronan is linked to the maturity of the sperm, the reduced risk of DNA fragmentation, reduced risk of aneuploidy, and better chromatin integrity. And the DNA fragmentation is an important message, because we know sperm DNA damage affects fertility. There's a whole range of studies showing that. But what we do know is the most compelling evidence that associates sperm DNA fragmentation with outcomes is the association with miscarriage or early pregnancy loss. So we can use hyaluronan binding to reduce the chance of picking a DNA damaged sperm, and therefore, hopefully, based on that evidence, we would reduce miscarriage, and I'll show you the data for that. Now, looking at the PIXI dish, this is a really nice paper from Yagchi and colleagues, where they labeled sperm with a fluorescent dye, and if they are red, they show DNA fragmentation, and if they're green, they are DNA intact. And what this showed is that when you put it on a PIXI dish, this is the PIXI dish with the hyaluronan dot, and what it shows really nicely is that outside the dot, so where they're not binding, around about 55% are labeled green, so their DNA intact. So 45% are DNA damaged. If you look inside the dot of hyaluronan, where the sperm are binding, what you can see is that 99.1% of the sperm are DNA intact. So you are drastically reducing the chance of picking a DNA damaged sperm if they're bound to hyaluronan on a PIXI dish. So does that translate into better outcomes? This is the Lodo Parmigiani paper from 2010. This is different because it uses sperm slope, but we saw reduced chance of picking DNA damaged sperm, not as effective as the 1% or so in the PIXI dish. But even then, it produced better quality embryos. So the grade 1 embryo quality on day 3 was significantly higher. Now, if you look at systematic reviews, this is one from 2014, which showed that hyaluronan selection didn't appear to improve clinical pregnancy rates, but note that they only had one study in each group. Similarly, if you look at live birth events, this is a later review from the same group, 2019, where they had 2 studies, including the HabSelect one, which I'll come back to. Now here, what's interesting is that it's still across the parity line, so it's not a significant improvement, but they concluded that there may be little or no difference between interventions, but if ICSI leads to a 25% live birth rate, hyaluronan ICSI, so PIXI, leads to a live birth rate ranging from 24 to 31. So it's definitely skewed towards producing a better live birth rate, but doesn't quite reach significance. But they did conclude there's evidence of a decreased rate of miscarriage in the intervention group. And that's important because that's the key finding, really. It backs up what Katie Warrilow found in a multi-center study from 2013, where they showed in men with poor hyaluronan binding, the miscarriage rate was drastically reduced, considerably reduced. And in the HabSelect study, so this is the paper by David Miller, which is a multi-center study across the UK of 2,500 patients. Again, what they showed was that there was an increase in live birth rate, but that didn't reach significance. But the miscarriage rate was improved, and statistically so. Now, if we look into that in a bit more detail, what we can see is, if you look at the underpinning biology, this is a nice paper from Champrou and colleagues from 2016, where they explored the mechanisms in oocytes to repair DNA damage in sperm post-fertilization. And what it really shows and explains really nicely is that all oocytes have some capacity to repair DNA damage. They recognize it and can repair it ahead of fertilization being completed. And where the oocyte quality is good, the outcomes are stabilized. But if the oocyte quality is poor, or the sperm DNA damage is very high level, excessive high level, then what they showed and explained is that you can get arrested DNA synthesis in the male pronucleus. That can impact embryo development, and where embryo development continues, you can end up with some degree of epigenetic changes and so on. Now that's interesting because it's borne out by this follow-up paper to the HabSelect study, which was from Western colleagues from Birmingham, published last year. And what this is showing is that, so this is looking at the same population that the Miller paper reported, but it's a mechanistic analysis. So they're looking for how and why PIXI might work. And they're taking a population that's enriched for miscarriage outcomes. And then what they're doing is they're looking at the impact of age on patients who become pregnant. So bear in mind, this is the predicted live birth rate once a woman becomes pregnant. And what you'll see here is you will see that the PIXI group, you see the fairly familiar age-related decline in live birth rate with age. So as the woman becomes older, we know there's a higher risk of the pregnancy terminating in a miscarriage. But in this mechanistic analysis, what they showed is that with PIXI, once the woman becomes pregnant, the likelihood of her carrying that term is not affected by age, or not as affected by age, which is why you end up with this straight line. So again, it highlights this story that by using PIXI, we avoid sperm DNA fragmentation. We know sperm DNA fragmentation is linked to miscarriage. If we use PIXI to avoid DNA damaged sperm, we can avoid the miscarriage, particularly in the older women where the egg quality is impacted and the repair of that DNA damage is compromised. They also showed an impact on male age, but of course male age has a much lower effect on overall outcomes, so that's there, but it's not as significant as the female age. So what this means is that if you get good quality oocytes and you do PIXI, the oocyte can repair the DNA damage, you get normal embryo development, and you get a normal chance of pregnancy. If you've got compromised oocyte quality, the poorer quality oocyte is less able to repair that DNA damage. You may end up with compromised embryo development. You may even get normal pregnancy rates, but you get an increased risk of miscarriage. And what PIXI can do is avoid that by avoiding the problem with the sperm. So quickly to summarize, sperm preparation should be performed in controlled conditions. We should handle the sperm in ways that minimize the impact on sperm DNA fragmentation. Centrifugation is probably okay, but we need to moderate the speeds we do it at where there's no evidence of harm. No particular sperm preparation method stands out as being better than others. Sperm prefer high bicarbonate and higher pH. PIXI, if you look in the data, gives variable results relating to fertilization, embryo quality and pregnancy, but there is this strong association with lowering the risk of miscarriage. And that association is probably indicating that PIXI is most effective when you target cases with high levels of DNA damage and advanced maternal age. So those are my selected references, and I'll end there and say thank you for listening.
Thank you so much, Dr. Dave. And I believe everyone was listening because we have few nice questions in the chat. Shall we go through them?
Yes.
So we have a question: is the composition of medias for sperm preparation important in case of PIXI?
I think, yes, I think it is. And I think what you can do is you can still use a medium that is more friendly to sperm. It will have less of an impact than it will in IVF or maybe even IUI. But I think it's good to put the sperm in a medium that is sperm friendly. What I would say is if for example, you were using the Origio medium and it's high pH, whether you're doing IVF or ICSI, you just have to take steps to make sure that you're not adding sperm and altering the pH to the egg. So if you put the sperm in PVP and allow the sperm to swim through the PVP, you can take them out and inject and there won't be a pH effect. For IVF, you'll need to put them into an IVF medium before you inseminate. What you don't want is large volumes of medium at pH 8 going into your insemination drop with your eggs at pH 7.3 because that might create a shift. Depends on the volumes you're using.
Yeah. We have another question. What about the clinical efficiency of chip-based sperm preparation techniques? Do they improve cycle outcomes?
I don't think that we can say that they do. They have some advantages in terms of workflow and efficiency potentially, but there is a paper in PLOS 1 that's just been published that has done a systematic review. And whilst there's a small trend towards improved outcomes with chips, it doesn't achieve statistical significance. And that's relating to clinical pregnancy rate and miscarriage rate. So it's worth people digging out that paper. It is literally appeared on LinkedIn yesterday, but they didn't show a statistical improvement in outcomes using a chip. As I say, there might be other advantages, but I don't think it improves we can say it improves outcomes.
All right. And we have a question about the pus cells sample. Does any of the sperm processing work with it? And how to deal with the ROS?
Sorry, what was the system?
The pus cells sample, does any of the sperm processing work with it?
What sort of sample? Sorry, Hadil, I'm struggling.
You can, okay, wait.
Pus cells. Okay, yeah. Okay. Yeah. Well, there's two different tacks to that. If you have pus cells in a sample and you have to prepare it, then a density gradient probably is the best approach. If you've genuinely got pus cells and you've got an active infection, you would possibly be well advised to treat the cause of those pus cells ahead of treatment if possible. By pus cells, I'm assuming they mean white blood cells, and white blood cells are usually transient if there's an active infection. So if that's there at the time of treatment, I'd be cautious about the way I handle that. But a density gradient should be able to remove those.
Okay, we have another question. To which group of patients would you recommend using the PIXI dishes?
Yep, I think, yeah, it's interesting. You can make an argument for using it for everybody because it reduces the chance of DNA fragmentation. But the real clinical benefit would be in the women over the age of, I think the West paper was 35 or 36. So the older women or men with persistent elevated DNA damage in their samples. So those are the ones that will benefit most. Mm-hmm. And that's what the mechanistic analysis in the West paper showed. And they suggested, and I'm raking through my memory now, but they suggested you could probably reduce the number of miscarriages by a significant proportion. I think it was around 40% by doing PIXI in those sort of groups.
How about the pH for the sperm preparation media? Is it important to measure it?
Only in as far as you would measure the pH of any other medium. I think that with the sperm prep medium, the heap is buffered, so the pH should be stable, and it's not important in the same way that measuring pH of your culture medium is, where the exposure time is a long time. For sperm prep, as long as it's in a target value, and the HEPs should control that. I personally wouldn't spend much time measuring pH of HEPs and MOPS buffered medium. I'd also say that you can't measure HEPs and MOPS buffered media if you're using some techniques like a blood gas analyzer, where the systems are calibrated to allow for bicarbonate.
Okay. We have a question. If you are aware of any studies that have tested the combination of microfluidics as a sperm selection and PIXI.
I think the quick answer to that question, Adil, is no. I can't, off the top of my head, no, I can't think of any. There may well be people doing it that haven't reported it. I just don't know of any studies that I could cite. What I would say is that I consider the microfluidic chips a way of selecting a population of sperm, and it's, they can be very effective at that, at producing a motile, morphologically normal sample with reduced DNA fragmentation, but it still leaves you with a choice at the time of ICSI about which individual sperm is injected, and only things like IMSI, PIXI, and Sperm Slow will allow you to do that.
Okay. Is density gradient really required compared to normal wash and swim up if normal samples?
You can choose whichever technique you want. The really important things to remember, I think, is if you choose to do a swim up, it's more difficult with the poor samples because the yield will be so low. Now, if you're only ever doing ICSI, that might be something you can tolerate. But if you're trying to do IVF, as you know, in some markets you have to do IVF, then a density gradient will always give you a better yield. So there are real advantages to a density gradient. The swim up as well, I think you have to be really careful that it appears to be such a simple technique, but if you wash then swim up, you're damaging the sperm. You have to do the swim up from whole semen, and then be careful with the harvesting. As I mentioned, there's that nice paper showing zinc as a proxy marker. If you do a swim up and you're not really careful with the technique, you get seminal plasma or components of seminal plasma in your final prep, which you want to avoid. It goes with the density gradient as well, that the attention to detail, getting the very clear interfaces is really important. So both of these look simple techniques, but the level of technical perfection needed to get the most out of it is really important to focus on. Very quick to do, but if you do it quickly and not well, you won't see the same results. So yeah, so coming back to the question, a density gradient you can use for every sample. A swim up is really only best used for those with good count and motility.
Okay, we have a question about the relationship between the size and speed of the sperm and sex. Some embryologists choose this method and they claim that they get more males in embryos like XY.
The only reliable way of testing the sex of sperm is to actually do fish on them, and there's all sorts of you can use density gradient methods to try and enrich male or female proportions of sperm. It's not an area I'm particularly well versed in, because in the UK we can't do it. So I've never really spent much time focusing on it. None of the techniques are, to my understanding, particularly robust. There are differences between X and Y sperm in terms of density and swimming speed and so on, but not so different that in a standard IVF lab you could rely on them as a way of improving sex selection.
Actually, we have plenty of questions. I'm not sure if we can take at least one more question.
Yeah, we can take one more question, and for the rest, we will try to share with you by email the answers for the rest of
I'm happy to try and email people with answers. Absolutely.
Perfect. So the last question we will take is, what about the lab environment and the impact on the sperm selection in terms of air quality, humidity, and temperature?
Yeah, good question. I wouldn't necessarily highlight sperm selection and sperm preparation. I think the air quality is applicable to all parts, and I think the overall message there is that whilst first of all you need to control air quality as much as possible, and the main concern in an IVF setting, so that includes sperm preparation, would be around control of volatile organic compounds. So we know particle counts we need to be wary of, but it seems to be the VOCs in particular that require more attention. So we need to focus on that. In terms of humidity, that certainly can have an impact on overall outcomes. I don't think it necessarily affects sperm particularly, but it can affect outcomes of the overall treatment.
For the rest, Amr, maybe we will reach out to them by email to answer the question.
Thank you, Dr.
And thank you everyone for all the questions. I can see there's lots of them, so it's nice to have that level of interaction.
Thank you so much.
Thank you everyone for joining and for taking the time today. We'll get back to you about the questions by email, and we're looking forward to see you in our next webinars. Have a great day. Thank you.
Thank you, everyone.